This brings us to the next part of our discussion, the bioorthogonal reactions. So, we will start with the basic introduction, then we will look at some of the types of reactions, and we will also look at applications. So, how we will move from chemical biology to bioorthogonal chemistry? We saw the limitations of some of the chemical biology reactions. So, we will move into the bioorthogonal condition. So, what is a bioorthogonal condition? Basically, cells or otherwise organisms are incubated with a metabolic precursor containing a unique functional group, that is biomolecule plus chemical entity are combined together. So, we had seen some of the requirement for reactions to be carried out under near neutral conditions and temperature around 40 degree or so, and this also should be very specific for a single functional group. So, these are some of the requirement, and the reacting the probe molecule bearing the complementary bioorthogonal functionality with the unique functional group. So, this is what when we talked about polymer beads or the probes, that is one part of the reagent where it is actually present, and the protein is the other part. So, connecting the probe with the protein is carried out by the chemical reaction. So, in the bioorthogonal reaction, we actually carry out the reaction between two functional groups, and this is going to be very specific for one particular functional group only. The first and the foremost reaction is the click chemistry. So, the click chemistry is used in both drug discovery and chemical biology. Of course, this reaction is now being used for bioorthogonal chemistry as well. So, what are click reactions? So, this is the mouse. So, in other words, you click, the reaction actually occurs. So, that is what is the basic definition for click reaction. So, click reactions actually is a reaction which is not a very specific single reaction. This is basically carried out by joining modular units by very simple reactions. So, two units are connected by very simple reactions, and this is very similar to how the nature works, and this name was coined by Barry Sharpless in 1998. So, this is the basic definition or what is a click reaction, and let us look at what are the characteristics of a click reaction. So, this is basically modular in nature, and this is having a wide applications or scope. So, this is one of the important reaction for any reaction to be considered a click reaction. So, as I mentioned in earlier slide, so click reaction is not a very single or a specific reaction. Click reaction is for any reaction which follows the characteristics as given in this particular slide. So, it will give generally a very high chemical yield, and this generally generates very inoffensive byproducts like water. Most of the reactions there will be no byproducts, and if the byproducts are going to be there, then they should be water or very similar non-toxic materials. And these reactions has to be very stereospecific and regiospecific. So, this is one of the requirement for click reaction, and it should be physiologically stable, and it exhibits a large thermodynamic driving force like more than 20 kilocalories per mole to favor a reaction with a single reaction product, and it also should have a high atom economy. So, these are all the characteristics of click reactions, and let us look at some of the examples of what are the click reactions. So, some of the examples include the 3+2 cycloaddition reactions or Huisgen 1,3 dipolar cycloaddition reactions, especially the copper 1 catalyzed azide alkyne cycloaddition reaction, or simply called as CUAAC reaction, and thiolene reaction which we had seen in the chemical biology system, Diels-Alder reactions and inverse electron-demand Diels-Alder reactions, 4+1 cycloaddition reaction between isocyanides and tetracenes, nucleophilic substitution on small strained rings like epoxy or the azides, and formation of urea. And we also have other carbonyl reactions like oxime formation and hydrazone formation. These are also some examples of click reactions, and photoclick reactions are also known. We will look at some of the details soon. The first one is the 3+2 cycloaddition reaction. This is the azide alkyne cycloaddition reaction. So, an azide and an alkyne are reacted in the presence of copper salt to give the triazole moiety as shown here. So, this is the very first copper click reaction, and the same reaction when reacted with the alkyne cyclooctyne, that is the cyclic alkyne is used, that is called the strain promoted azide alkyne cyclization reaction. So, this is a very strained ring with the alkyne unit in the 8-membered ring, and this undergoes a very efficient click reactions when treated with the Azide unit. So, this is how the reaction actually proceeds. So, these two are examples of 3+2 addition cycloaddition reactions. So, the 3 unit is here and the 2 unit is from the alkyne unit. The next one is the inverse electron demand Diels-Alder reaction. So, here an alkene is reacted with the tetrazine. So, this is a inverse Diels-Alder reaction with the loss of nitrogen. These 2 nitrogen are lost as a nitrogen gas, and this is a place where the alkene undergoes addition to give the condensed product as shown here. And another one is the Schrodinger ligation, which we saw an Azide when treated with the triphenyl phosphine derivatives leads to the formation of the condensed product as shown here. So, this is also called as Schrodinger-Betrose ligation reaction. And I told a little bit earlier like a ketone condensation reaction. So, we saw some examples. So, here in this particular case, either hydroxylamine or hydrazine is reacted with the carbonyl compound, and the reaction generally is carried out between the pH 4 to 6 to give the corresponding oximes or hydrazone derivatives. So, this is also another example of a click reaction, and we also have 4+2 cycloaddition reaction as shown here. Another set of reactions using thiol is 1,2-aminothiol and the 2-CBT, that is 2-cyanobenzothiazole. So, these two undergo condensation reaction to give the corresponding condensed product as shown here. And we saw earlier strain promoted Azide, and so instead of an Azide we have a nitrone here. So, the strain promoted alkyne nitrone cycloaddition reaction is shown here. So, we end up with the condensed product as shown here. So, this is also another example of click reaction. And the last one is the photoclick reaction, or the photoclick 1,3 dipolar addition reaction. So, here a tetrazole is reacted with an alkene in the presence of photo conditions with the loss of nitrogen molecule to give this particular derivatives. So, these are some of the examples of click reactions. There are various bioorthogonal reactions which are possible at ketone and aldehyde. Some of them we had already seen. The first one is the oxime reaction, the next one is the hydrazone formation. So, these two are you are already familiar with. So, these are all the 2 reactions. Other than that, we also have other reactions with aldehyde and ketone substrates. So, the Pictet-Spengler reaction which is giving a cyclic product as shown here, or the Pictet-Spengler ligation in which the X group is oxygen is also known, and the Wittig reaction of a Wittig salt with carbonyl compound to give the olefinic product. So, these are some of the examples of bioorthogonal reactions which can be carried out at ketone or aldehydes. And bioorthogonal reactions with acids, we have seen one of the example, that is the cyclooctyne condensed strain promoted azide alkyne cyclization reaction. Other than that, we also have a few other reactions. The Schrodinger ligation uses the triphenyl phosphine as one of the substrate, and we also have phosphite and phosphinite reactions. So, these two also leads to the condensation reaction. So, these are all the different types of bioorthogonal reactions which can be carried out and the Azide. And in the case of alkynes, the alkyne can be a straight chain alkyne or acyclic alkyne, another one is a cyclic alkyne. So, these two reactions can also be carried out. So, the alkyne can undergo reaction with the thiol. So, this reaction is very similar to thiol-ine reaction, or this is called as thiol-ine reaction, which is very similar to thiol-ene reactions. And the azide alkyne cyclization we already know, and the tetrazine also can be used, or syndones can be used. So, these two are additional reactions which reacts with the alkynes, especially the cyclic alkynes, to give the condensed product as shown here. So, we can also introduce alkynes to the metabolic systems. So, there are various examples are given here, and it can be the addition of only 1 or 2 atoms, or it can be an entire molecule. So, depending on, but in all the cases what happens is the alkyne is actually incorporated into either DNA, so as shown here, or to the RNA. So, these are all the modifications which has to be carried out on the system. The biological system is modified using these kind of alkynes at a particular site. So, unlike the previous cases where if we are talking about proteins, the proteins have multiple sites for modification. So, we use a very specific amino acid to be incorporated into the protein structure or the RNA structure or a DNA structure. So, this is the major difference between simple chemical biology reactions and the bioorthogonal reactions. So, in the chemical biology reactions, we are actually choosing the system. Basically, the protein or the RNA or the DNA will have a particular functional group, and the reactions are carried out on that particular functional group, like amines or thiols or similar groups. But unfortunately, the protein or other groups have more than one reactive place. So, that means we cannot be site specific labeling is going to be a very difficult challenge to carry out. Whereas, when we do the bioorthogonal reactions, what we are doing is we are actually modifying the biomolecules with one of the functional group. Once the functional group is incorporated into the biological system, then we can use the second unit, which is the probe, which will exactly go and add or undergo reaction only at a single site. So, if we are going to introduce alkyne unit into the biological system, this particular area is the only place where the say for example, if we do the copper azide alkyne cycloaddition reaction, or the previous one what we have seen, any of this azide alkyne reaction like a thiol reaction or the tetrazine reaction, these kind of reaction will be happening at only one particular site. So, it will not be like the entire protein or the phospholipid or the triglyceride will not be affected. Only the particular site which is our interest will be modified. So, these are all the advantages of using bioorthogonal reaction. And in situ click chemistry can also be used for enzyme inhibitors. So, the active target can be condensed as shown here. So, as the green one, the bioorthogonally reactive groups can be used to condense either reversibly or irreversibly to form a covalent bond, or bidentate ligand can also be used. So, these are some of the ways by which we can actually couple two things together. So, this line is the one which is a linker which connects the building blocks or the fragments to our bioorthogonal reactive functionalities. So, this is the crucial thing. So, these two are connected at two different places, and when a reaction takes place under biological system, these two undergo reaction very effectively. There are some functional groups which can be used for these kind of conjugations. We can use a carboxylate unit, or we can use aldehyde unit, thiols, hydrogens, ketones, hydroxy groups, amines, etc. on the biological system can be used for modification. Let me discuss about our work, what we have done in the bioorthogonal chemistry. So, this work actually appeared in the journal cover page of Chemical Communication. So, we have carried out a bioorthogonal reaction for molecular and cellular imaging. A strain promoted alkyne azide cycloaddition reaction, spark of a novel EPCAM aptamer fluorescent conjugate for imaging of cancer cells. So, this is the work we have carried out. This appeared a few years back in the ChemCom cover page. So, what is the reaction we actually carried out? Let us go back to what we have seen, the bioorthogonal reactions at alkyne and the cyclooctynes. So, the reaction what we have focused was cyclooctyne and the azide condensation reaction. So, this is the click reaction which we had actually focused on. So, the azide and the octyne are condensed together in the presence of a copper catalyst. That is a standard reaction, and in our case what we carried out was there was no copper catalyst used because this is a strain promoted reaction. So, there is a extremely high strain because of the cyclic system, As you know, the alkyne is basically a sp hybridized carbon. So, the sp hybridized carbon actually is this is basically the linear geometry of sp hybridized carbons, and when a linear geometry is placed in a cyclic system, this actually leads to extremely high strain. So, the ring strain is going to be very high in these kind of cyclic systems, and to relieve that, what happens is the reaction, the cyclic alkyne cannot exist for longer time. So, this has to undergo some reaction, and the reaction here is the formation of the bicyclic system. So, this is basically the alkyne, which is a sp hybridized carbon, is converted to a sp2 hybridized carbon, and when it is converted into an sp2 hybridized carbon, then that sp2 hybridized carbon can exist in the cyclic system comfortably. And this is one of the steric strain is responsible for this particular efficient reaction to take place, and this is the one we have actually focused on, and we thought this is going to be a very good reaction for the condensation, and we chose this for the bioorthogonal reaction for our studies. So, as I mentioned, this is a strain promoted azide alkyne cycloaddition reaction. So, this is a 1,3 dipolar cycloaddition of azide with terminal alkynes to produce 1,4 disubstituted 1,2,3 triazoles. Here we do not have a terminal one, we have a cyclic system here, and when the reaction is carried out in the presence of copper 1 catalyst, this cycloaddition reaction proceeds extremely fast, and this avoids regioisomers. So, this is one of the major difference between using copper catalyst and without copper catalyst. When we are not using a copper catalyst, we actually end up with regioisomers, whereas when we are using a copper catalyst, we do not end up with a regioisomer. So, our work was basically couple of steps were known, couple of steps were not known. So, how we did, I will just explain. So, the bioorthogonal reactions or the spark can target a biomolecule. So, this is already known. The EPCAM is overexpressed in various cancers. EPCAM aptamer is used for cancer targeting. So, this is also known. So, although these two things are known, what is the missing link is aptamer based bioorthogonal reactions targeting EPCAM positive cancers are not known. And of course, we were one of the first group to identify this kind of aptamer based bioorthogonal reaction. Although bioorthogonal reactions are known in the protein field and other areas, in the aptamer area it was not highly known. So, we have a aptamer sequence which is known for EPCAM specificity. This is a probe which we actually identified, and this aptamer sequence, and we have to add a specific functional group to the probe. So, that means Azide group has to be incorporated on this EPCAM aptamer. And Alexafluor is basically the fluorescent dye, as I mentioned earlier. So, we need a probe, and the probe generally has a fluorescent label, and the fluorescent dye what we have used is Alexafluor 594, and DIBO is the octane derivative which is used for our imaging studies. So, the aptamer has the Azide unit, the DIBO is having the fluorescent probe, and this is having the alkyne unit. So, when these two are combined together, this gives the triazole derivative. So, this is what our overall work. So, this Azide aptamer is one unit, and the alkyne DIBO and the AF 554, the Alexafluor is another unit, and these two are condensed together to give the click reaction. So, the structure of the compounds are given here. So, the DIBO structure is dibenzooctane, cyclooctyne is the one. So, we have the two benzo unit, one benzo unit here, another benzo unit here. So, these dibenzo units are used on the octane unit, and this is the Alexafluor which is attached to this particular octane, and this is the aptamer unit, and this aptamer unit is having the Azide derivative. So, these two are condensed together in the presence of copper salt to give the Azide alkyne condensed product as shown here. So, the triazole conjugate was formed. And when we use 1:1 ratio or 1:2 ratio of the azide and the alkyne, the reactions are not very high. So, when we use 1:6 ratio, we end up with our final product in a very high quantity. And if we just use pure Dibo AF4, that is fluorophore, this shows very high on the TLC, whereas in the gel electrophoresis, whereas when we use 1:1 ratio, the reaction is very small. So, this is one of the reason we have to use a little bit higher ratio of the reacting substances. And once the reaction actually happens, the fluorophore is actually going to be attached to the EPCAM on the cell surface, and we can actually see the where this particular unit is attached, that is the EPCAM aptamer, which part it is attached, whether it is on the cell surface or it is in the nucleus. So, these two molecular imaging we have carried out later to identify where is actually the EPCAM aptamer present. So, by that, because this particular EPCAM aptamer binds to the cellular receptors which are overexpressed in cancer cells. So, once the conjugate was formed, we did the HPLC isolation, and the conjugate is shown here as a single product, and the purification gave the aptamer alone has a molecular weight as shown here using MALDI TOF, and the aptamer-conjugate with the azide alkyne conjugate is having a higher molecular weight. So, this confirms that the product is formed. And then we carried out some stability studies, because as I mentioned, when we are carrying out bioorthogonal reaction, the reaction or the product has to be stable under physiological conditions. So, this compound was intact up to 72 hours according to agarose cell electrophoresis, and the dialysis also did not show any degradation or release of this particular part, Dibo. So, what we did was we connected the Dibo with the aptamer. So, it can break in between these two things, because these two are connected by simply the condensed reaction or the triazole formation. So, if the triazole is unstable under physiological condition, then our study cannot actually lead to the required results. So, we just wanted to make sure these conjugates are quite stable under physiological conditions. And there are two types of aptamer interactions are possible. One is a membrane bound one, because this EPCAM aptamer is overexpressed in the cellular and the nucleolin based one. So, we wanted to know whether the aptamer is going to interact with the membrane, or it is going to be the internalized one, or whether it is going to the nucleus. So, to study that, we carried out the conjugations at incubation at two different temperatures. So, for the membrane bound, we carried out the reaction at 4 degree, and for the internalization studies, we carried out the reaction at 37 degree centigrade. And the 4 degree centigrade reaction was studied by flow cytometry, and the internalization was studied using fluorescent microscopy. So, these two are two different types of analytical techniques which actually tells where is our aptamer. So, the flow cytometry analysis of basically the different cell lines what are used are shown here. The first one is the control, the second one is Dibo alone, and the third one is our DNA aptamer with the Dibo conjugation. So, if you look at the mean fluorescence intensity, we are having a very high intensity for our conjugated species. So, this tells very clearly that our reaction actually are targeting the required space. And the flow cytometric analysis also had shown that see, this is the MCF7 conjugate, and this is MCF Dibo and AF the conjugate, which you can see the very high intensity here, and the MCF alone or the unstrained one is not having high intensity as shown here. So, these are some of the experiments which shows that the self surface binding of our aptamer was confirmed. And when we carried out inverted fluorescent microscopy, some of the images are shown here. So, the upper panel is the one with the DAPI and AF594 merged, and the lower panel is showing the respective phase images. So, this actually you can see the color here. and these colors actually show that the aptamer conjugate are actually bound to the cell surface. Here we have the inverted fluorescent microscopy images of the aptamer conjugate. So, this is one of the retinoblastoma cells. So, here we actually see when we use the conjugate, you can see the red color at the periphery of this one. So, this actually tells about the cellular uptake, and this is a negative one. So, here you do not see the uptake. So, this clearly tells that whenever there is a positive cells, then we actually see the cellular uptake at the cell surface. And we have other two cases. This is a dark field fluorescent microscopy, the MCF7 cells and the conjugates, and very RB1 is retinoblastoma cell and the conjugate. So, you can see the red color very clearly. So, these are all the examples which clearly tells that our click reaction worked fine, and whatever the aptamer which we are using are targeting the required space. And we also had shown with the other type of cells as shown here. When we did the cytotoxicity of the aptamer conjugates, the control is having a very high cytotoxicity, whereas some of our obtained conjugates are not showing very high cytotoxicity after long hours. And we also had confirmed that serum stability of our bioconjugates. So, this shows that our conjugates are stable up to 24 hours and they show no significant degradation, and up to 24 hours, but when we go to 36 hours there is a degradation of 70%. So, this tells that after 1.5 days the material can be actually removed from the system. And if we actually want to keep our conjugate at longer duration within the body, we may have to do some suitable modifications. So, with that I conclude the reaction, the bioorthogonal reactions, introduction and strategies. Thank you.